Assistant Professor of Geology, to welcome the gathering and to introduce the resource person of this session, Dr. S. Manivannan, Assistant Professor of Geology. Madam, please.
Am I audible, sir?
Yes, madam. You can proceed, madam.
Okay, thank you. Good morning to one and all present here for the fifth day of webinar series on Vistas in Research organized by Kongunadu Arts and Science College, Coimbatore. It gives me great pleasure to extend a very warm welcome to our Honorable Secretary Madam, most respected Principal Sir, beloved Chief Executive Officer Sir, Dean, Research and Development, and Dean Academics. I also extend my warm welcome to our guest of honor, Dr. S. Manivannan, Assistant Professor, Department of Zoology, Kongunadu Arts and Science College, Coimbatore, who is with us today to share his expertise on infectious diseases, immune response to infection. And also, I wish to welcome the faculty members and the students of Kongunadu Arts and Science College and other academic and research institutions. I also take this opportunity to welcome and thank our organizing team for organizing such an informative webinar, which gives an opportune time to renew contacts and discuss problems of mutual interest with delegate. The agenda of the webinar covers a wide range of very interesting topics. I once again, on behalf of the Kongunadu Arts and Science College and on my own, I welcome you all to the fifth day of the webinar series on visitors in research. Before we start the session, I wish to share with you all the credits of this session presenter, Dr. S. Manivannan. Currently, he is working as an assistant professor in the Department of Zoology, Kongunadu Arts and Science College, Coimbatore. After he got graduated in zoology from Bharatidasan University, he got qualified CSER examination in the year 2002. Then he joined his PhD in the Clinical Pathology Laboratory, National Institute for Research in Tuberculosis, Indian Council for Medical Research, Chennai, where he got expertise in the field of infectious diseases. Later, he did his postdoc at Department of Biotechnology, Madurai Kamaraj University with DBT postdoctoral fellowship. He has more than 7 years of teaching experience and 18 years of research experience. To his credit, he has published 25 research articles in refereed journals and one review article and one book. He is also a recognized guide and currently guiding 5 PhD students and completed 16 MSc theses and 7 MPhil theses. With this introduction, I would like to say that during these pandemic days, we all have heard a lot about the immunity, and our immune response is the only way by which we can beat the COVID-19. Still, many of us having the curiosity that how our immune system is responding to the pathogen and how it helps us to get rid of from the pathogens. I hope at the end of this presentation, we all will get the answer for our curiosity. I wish everyone to have a great time with us today. Now I hand over the session to Dr. S. Manivannan to share his expertise with us. Please, sir. Thank you.
Thank you, madam, for your nice introduction.
Thank you, sir.
Thank you.
Thank you, Bharati Madam, for your nice introduction. Good morning to one and all present here. The great honor for me to welcome once again Honorable Secretary Madam, respected Principal Sir, Dean, Research and Development, and Academic Dean, and the most valued participants of webinar series on Vistas in Research organized by Congrado Arts and Science College, Coimbatore. I wish to extend my heartfelt thanks to organizer for offering me this opportunity. I will start my talk with the two quotes. The first of all, in Tirukural, Noi Nadi, Noi Mudal Nadi, Adudanikum Vai Nadi, Paipachayal. The second quote proposed by William James, that is, the glory of medicine is, that is, to the constantly moving forward, that there is always more to learn. The ill of today do not cloud the horizon of tomorrow, but act as spur to great effort. To begin with, let me ask you all the basic question. All the basic question that what is infectious disease? Okay, that's great. The non-biologists are also participating in this webinar. So let me start with the basics. Infectious disease are disorders that are caused by various organisms, usually microscopic in size, such as bacteria, virus, fungi, or parasites that are passed directly or indirectly from one person to another. Humans can also become infected following exposure to an infected animals that harbor a pathogenic organism that is capable of infecting humans. Next, if you see the infectious disease or the one which leads to cause the death worldwide, particularly how in the low income countries and particularly in the young children, the infectious disease are associated mortality and are remaining a significant threat throughout the world. In the 21st century, we continue to fight both world pathogens like plague that have afflicted humanity for millennia and new pathogens like human immunodeficiency virus and COVID-19 that have mutated or spilled over from animal reservoir. If you see the pandemic of infectious disease, what is pandemic? The pandemic are the disease which is prevalent throughout the world, but if you see the epidemic or disease which are confined to a particular regime, this picture depicts the pandemic of various infectious disease since Antawan plague in the 2nd century to current COVID-19 even. For example, Antawan plague or outbreak in the 2nd century and bubonic plague in the 14th century, smallpox in the 16th century and the Great Plague in the 17th and 18th century. In cholera and the Spanish flu and Russian flu, the pandemic to the 19th century, HIV and AIDS in 20th century, SARS, severe acute respiratory syndrome, MERS, the Middle East respiratory syndrome, and COVID-19 in the 21st century. Most of the pandemic are come from the Chinese and the Hong Kong region. For example, the malaria, tuberculosis, leprosy, influenza, smallpox, and others first appeared during these early years. After the more civilized, after the in between the 2nd and 13th century, there is no large outbreaks because of the more civilized human become with the large cities, more exotic trade routes, and increased contact with the different population of people, animals, and ecosystem, the more likely pandemic would occur. According to WHO in 2017, the infectious disease are ranked in the top 10 causes of death worldwide. They are lower respiratory infections, more than 3 million deaths, the diarrheal disease, 1.1 million death, and tuberculosis, 1 million death. HIV and AIDS infection has dropped from causes of death 1 million deaths in 2016 compared with 1.5 million in 2000. But it is still a leading cause of death in undeveloped and developing countries. Another infectious disease, malaria, accounts for a top cause of death in undeveloped countries, especially in African continents. So far, we are discussing about the pandemic of disease. Now let's see how above said diseases are prevented by vaccination. So, vaccination is one of the most effective way to prevent the disease. A vaccine helps the body immune system to recognize and fight the pathogen. A vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. The routine immunization programs protect most of the world children from a number of infectious disease that previously caused millions of deaths each year. So far, we have used vaccines to entirely wipe out two diseases, smallpox and rinderpest, which infected cattle. We have also come extremely close to eradicating polio, which less than 500 new cases annually, largely in Afghanistan, Nigeria, and Pakistan. In the United States, a long list of diseases like have been nearly eradicated by vaccine, diphtheria, bacterial influenza, and measles, mumps, rubella, and tetanus, among others. Still, there are the many developing countries that have the limited vaccine supplies that scanned funding for childhood vaccination service, which has allowed them preventable diseases like whooping cough and rotavirus to continue spreading. However, the satisfactory vaccine have not yet been developed against the several of most life-threatening conditions like COVID-19. There are 5 different stages which leads to the endemic human disease. Where did they come from? Why are they overwhelming of world? The major human infected disease have animal origin, and we continue to be bombarded by novel animal pathogens, yet there is no ongoing systematic global effort to monitor for pathogens emerging from animals to human. Such an effort could help us to describe the diversity of microbial agents to which our species exposed, to characterize animal pathogen that might threaten us the future, and perhaps to detect and control a local human emergence before it has a chance to spread globally. Next, the challenges in infectious disease research. The first one, the new infectious disease emerged to continue. Since the 1970s, about 40 infectious diseases have been discovered, including the SARS, MERS, Ebola, chikungunya, avian flu, swine flu, and most recently, Zika, hantavirus, and COVID-19, and etc. The second point, the old infectious disease increase in incidence or the geographical distribution and previously under control to re-emerge. The re-emerging diseases include malaria, tuberculosis, cholera, pertussis, influenza, and pneumococcal disease and gonorrhea. The third challenge is the intentional introduction of infectious agents by bioterrorists. The release of biological agents like viruses, bacteria, fungi, or toxins. For example, the anthrax, botulism toxin, plague, viral hemorrhagic fever, and etc. The fourth challenge, the increasing resistance of pathogen to current antimicrobial drugs. In 2016, 490,000 people developed multi-drug resistant TB globally, and drug resistance is starting to complicate the fight against the HIV and malaria as well. The fifth challenge, the breakdown in public health system and communication between nations. Next, the different classification of infectious disease. The infectious diseases are classified based on the duration of the exposure and the type of causative agents. The further, the disease caused by agents are classified into communicable disease and non-communicable disease. Then, the disease are also classified based on the causes of disease and the medium from where the pathogens arrived. Next, the human infectious disease and their causative agents. There are 5 major types of infectious agent: bacteria, virus, fungi, protozoan, and helminths. In addition, a new class of infectious agent, the prions, have recently been recognized. Next, the mode of infectious disease and transmission. There are a number of different routes by which a person can become infected with an infectious agent. For some agents, human must come in direct contact with a resource of infection, such as contaminated food, water, fecal material, body fluid, or animal product. With other agents, infection can be transmitted through the air. The route of transmission of infectious agent is clearly an important factor in how quickly an infectious agent can spread through the population. An agent that can spread through the air has greater potential for infecting a large number of individuals than an agent that is spread through direct contact. The another important factor in transmission is the survival time of infectious agent in the environment. An agent that survive only a few seconds between the host will not able to infect as many people as an agent that can survive in the environment for hours, days, or even longer. These factors are important consideration when evaluating the risk of potential bioterrorism agent. Next, how infectious agent cause the disease. Infection with a pathogen does not necessarily lead to disease. Infection occurs when virus, bacteria, or other microbes enter our body and begin to multiply as a result of infection. Sign and symptom of an illness will appear. Let's discuss toxin and the enzyme produced by microbes and its impact in the cell. When the microbes enter into our cell, it produces a toxin which in turn promote infection and disease by directly damaging the host tissue and by disabling the immune system. For example, the Clostridium tetani produce the tetanus toxin which leads to a fatal condition known as tetanus in many vertebrates including humans and invertebrates. Then many pathogens produce a series of enzymes to help overcome body defense and establish themselves in the host. One example, in leukocidins, a group of enzymes that destroy white blood cell, this destruction lessen the body ability to perform phagocytosis. Next, moving to the phases of infectious diseases. When a pathogen enter into a cell, it undergo the 5 stages. The stage 1, the incubation period, stage 2, prodromal phase, stage 3, clinical phase, and stage 4, decline phase, and stage 5, recovery phase. The stage 1, incubation period, the time between the infection and the appearance of sign and symptoms. The stage 2, prodromal phase, a mild non-specific symptoms that signal onset of some disease. The third stage, the clinical phase, a person experiences typical sign and symptom of disease. The fourth phase, decline phase, a subsidence or reduce of symptoms. The fifth and final stage, recovery phase, the symptoms have disappeared, the tissues heal and the body regains strength. The next moving to outcome and exposure to an infectious agent. There is a spectrum of possible outcomes following the exposure to an infectious agent. Some person exposed to pathogenic microorganism never develop a systematic disease, while others become severely ill and when die. Next, move on to the chain of infection. This infectious disease could be overcome by breaking the chain because the chain of infection is fundamental to infection prevention and control. All the component of chain of infection must be presented for an infection to occur. If one of the link in the chain is missing or is deliberately broken, the spread of disease to another is effectively controlled. Consider if you avoid contacting the reservoir for quite a time, the pathogen will get extinct, by which we can break the chain, most important way by which we can break the chain is hand hygiene is the single most effective way to break the chain of infection, which are following the break the chain of COVID-19 to overcome the outbreak of pandemic of COVID-19. Next, moving on, the disease triangular. In addition to all the above mentioned point, infectious diseases are not causing the disease only by the entry of pathogen. It also depending on the host susceptibility and the environment. These three elements, the pathogen, host, and environmental conditions, make up the disease triangle. The disease triangle is a concept that illustrates the importance of all the three elements, just as there are the three sides to a triangle. There are three critical factors necessary for a disease to develop. For example, in India, we are not very much prone to COVID-19. It is because of rate of your susceptibility and the environment pre-existing immune response. Next, move on. The mechanism of pathogenicity. Although various pathogenic microorganisms can be responsible for many different disease syndromes on host of all kinds, several factors are common to the establishment of infection. The first step is primarily oriented towards the outer surface of host and involves the adhesion of the microorganism to host cells or tissue. Many pathogenic microorganisms have been shown to utilize the specialized surface associated adherence factor or adhesion to attach themselves firmly to their host. After attachment, the pathogenic microorganism may physically penetrate or enter the host. The next stage, the microorganism need to colonize the host in order to feed and replicate. The mode of infection of such pathogenic microorganism is somewhat unsophisticated, as they apparently do not avoid triggering the host defense system and tend the adapter to kill host cell as quickly as possible. This situation actually resembles human and animal infection by microbial pathogens. Let's move on the transmission and dissemination of microbes. When the microbes invade into our body, it is not only targeting the particular tissue, it will transmit into other organs as well. For example, everyone says that COVID-19 transmit to the lungs and it causes inflammation in the lungs. But recent research published by the scientists from England highlighted that COVID-19 is also cause inflammation in the intestine and also the soft tissue. So far, we are discussing that the basics of pathogen, route of entry, and dissemination of pathogen to different organ system. Is the second part. The first part is what are the infectious diseases, and the second part, what is immune system and how our immune system defend the infection. Now, we are moving to how our immune system is responding to the pathogen when it encounters our body. Could anyone say what is immune system? Yes, the immune system is made up of different organs, cells, and proteins that work together to fight against the pathogen enter into our body. That is the one which keep us healthy, though we are exposed to various pathogens nowadays. Yet, still we have alive and not getting sick. It's all because of your amazing immune system. At this point, one point we should all remember that some infection like the flu and the common cold have to be fought many times because so many different viruses or strains of same type of viruses can cause this illness. Catching a cold or flu from one virus does not give you immunity against the others. Next, moving on, overview of immune process. If you see the overall immune process in our body, 4 main processes in taking place like recognition, checking, acting, and regulating these are regulated by various cells and genes. Next, the genes related to the type of immune response. Before we go in detail about the immune response, let us briefly see genes responsible for immune response. Among the genes responsible for the immune responses, 27% of genes are responsible for innate immunity, 41% are adaptive immunity, and 32% responsible for both immunity. Many participants may hear the word innate and adaptive immunity in the first time. I will explain later. Next, to move on. The innate immune system is an older evolutionary defense strategy, relatively speaking, and is the dominant immune system response found in plants, fungi, insect, and primitive multicellular organism. Adaptive immune system is also referred as the acquired immune system and is found only in vertebrates. The next to move on, the evolution of immune system, particularly in the vertebrate. The most sophisticated immune system are those of vertebrates, recognizably lymphocyte and immunoglobulin. The the immunoglobulin, otherwise called the antibodies, appear only in this organism. The most primitive living vertebrates, the jawless fishes, example hagfish and lampreys, do not have the lymphoid tissue corresponding to a spleen or thymus, and their immune responses, although demonstrable, are very weak and sluggish. Further up, the evolutionarity at the level of cartilaginous fishes, shark and rays, and the bony fishes, the thymus and spleens are present as there are immunoglobulin, although only those immunoglobulin of IgM classes are detectable. The fish lack specialized lymph node, but they do not clusters of lymphocyte in the gut that they serve an analogous purposes. Though all the vertebrates having the immune system, but only mammals are the group of living organism which has the well-developed immune system. Next, move on, the schematic figure of the postnatal development of the innate immune system. If you see the systematic figure of the postnatal development of the innate immune system, the length of the elongated arrows indicate the time point at which the full functional capacity is obtained and no further changes apparently occur in basic innate immune components. According to the current literature, neonatal immunity is often characterized as immature, mostly due to impaired adaptive responses in comparison to adult, especially concerning the deficiencies in B cell function and antibody production, but also the limited in magnitude of T cell responses and their partial bias to type 2 response. What is the postnatal innate immune development from birth to adult? It is well established that the adaptive immune responses are deficient in early life, contributing to increasing a mortality and morbidity. The developmental trajectories of different components of innate immunity are only recently being explored. Next, the comparative analysis of immunoglobulin genes. If you compare the genes for the different immunoglobulin, only the mammals have all the immunoglobulin gene. They contain the 5 immunoglobulin group: IgG, IgM, IgA, IgD, and IgA. The other group of animals are lacking one or the other gene. For example, amphibians have only IgM, IgY, and IgX. Next, the genetic origin of immunoglobulin. And if you see the genetic origin of the immunoglobulin, this schematic picture shows that some proteins of the immunoglobulin superfamily are involved in the cellular ability to organize other cells or foreign particles. They share a basic structural similarity in the so-called Ig domain. Show added in the blue, indicating that the gene encoding this protein evolved from common ancestral gene involved in cell-to-cell recognition. The next to see the hematopoietic stem cell derived the cell lineage. The cells responsible for this immune response, the pluripotent hematopoietic stem cell, differentiate in bone marrow into the common lymphoid or common myeloid progenitor cells. The lymphoid stem cell give rise to B cell, T cell, the natural killer cell lineage. The myeloid stem cell give rise to a second level of lineage specific colony from unit CFU, cells that go on to produce neutrophil, monocyte, eosinophil, and basophil and mast cell, megakaryocyte and erythrocyte. The monocyte differentiate further into the macrophages and the dendritic cell. The dendritic cell are the one which responsible for the adaptive immune response and hence the function as the sentinels of the immune system. Recent research stated that new type of dendritic cell have been identified. This will give us some insight to fight against the COVID-19. Next, moving on. Most of us are aware of the different type of the blood cell and its roles. Next, the phagocytic cells of the innate immune system. The phagocyte are a type of white blood cell that use the phagocytosis. What is phagocytosis? The phagocytosis engulf the bacteria or the foreign particle and the dying cell to protect the body. They bind to pathogens and internalize them in phagosome, which acidifies and fuse with the lysosome in order to destroy the content. They are a key component of innate immune system. They are three main group of phagocytos: monocyte, macrophage, granulocyte, and a dendritic cell. Many cells are capable of phagocytosis. Example, the first one, neutrophil. Neutrophil, these are abundant in our blood, nearly 40 to 60%. The important role of neutrophil is acute inflammation, as they are the first immune cell to arrive at the site of infection. Second cell is macrophages. The macrophages, nearly the 5 to 10% of our blood, that are the key as a first defense mechanism and in initiating the adaptive immune response. Third one, the dendritic cell. The dendritic cell, they cycle through the bloodstream, tissues and lymphoid organ, sampling potential pathogen and acting as a major link between the innate and adaptive immune system. Next to move on, now we are going into detail that how our immune system is responding. As I said earlier, there are the two different type of immune response. One is innate immune system and another one is the adaptive immune system. The innate immune system means the inbuilt or from parents or from the genes. The acquired immune system to acquire from the outside of the body. The innate immune system is a non-specific and this will not keep any memory. In adaptive immunity is a specific, it keeps a memory. Next, moving to them, two type of immune system, one innate immune system and adaptive immune system. Just as resistant to disease can be innate or inborn or acquired. The mechanism mediating it can be correspondingly divided into innate immunity on the left hand side and adaptive immunity on the right hand side. Each composed of both cellular level in the lower half and the humoral element in the upper half. The cellular elements is our system, our immune system contains the different type of cells like the lymphocyte, the 5 different type of lymphocyte and the monocyte and neutrophil, eosinophil and basophil, and other cell type of cell means B cell and T cells. In the other immune system is humoral immunity. Humoral immunity means antibody-mediated immunity. These immunity perform many of their function by interacting with the older innate ones. Next, explain on adaptive immunity. The adaptive immunity is defined by two important characteristics, specificity and memory. Specificity refers the adaptive immune system's ability to target the specific pathogen, and the memory refers to the ability to quickly respond to the pathogen to which it has previously been exposed. For example, when an individual recovers from chickenpox, the body develops a memory of infection that will specifically protect it from the causative agent, the verticillular zoster virus, if it is exposed to the virus again later. Next, if you see the innate and adaptive immune timeline, the mechanism of innate immunity provides the initial defense against the infection. The example, innate immunity start at the 0 to within the 12 hours. The adaptive immune response develop later and require the activation of lymphocyte. The kinetics of the innate and adaptive immune response are approximation and they may vary in the different infection. Next, moving on, the barrier defense mechanism of innate immune response. If barrier defense are part of the body's most basic defense mechanism, the barrier defense are not a response to infection, but they are continuously working to protect against a broad range of pathogens. The primary barrier to the entrance of microorganisms into the body is skin. The innate immune response usually begin with the physical barrier that prevent the pathogens from entering the body, destroy them after they enter them out before they can establish themselves in the body's soft tissue. The barrier defense are a part of body's most basic defense mechanism. The barrier defenses are not response to infection, but they are continuously working to protect against a broad range of pathogen. The primary barrier to entrance of microorganism into a body is the skin. Another barrier is the saliva in the mouth, which is rich in lysozyme, an enzyme that destroy bacteria by digesting their cell wall. The acidic environment of stomach, which is fatal to many pathogens, is also a barrier. Additionally, the mucous layer of gastrointestinal tract, respiratory tract, reproductive tract, eyes, ear, and nose traps both microbes and debris and facilitate their removal. Next, moving on, soluble mediators immune system. Our immune system or our blood contain the two different type of cell. One is called the cell, different type of cells, and other one is called the soluble matters. So far, we were discussing about the immune cell, the genes responsible and the evolution, the immune response. Now we are moving to molecules which are produced against the pathogen. These are four soluble mediators of the immune system. One is antibodies, second one complement, third one is cytokine, and the fourth is inflammatory mediators. I already explained there are the antibodies, the 5 different type of antibodies present in our serum. The second one is called the complement. What is the complement is additional to the antibodies, that is called the complement. The complement contain the more than 25 different type of proteins in our blood system. What is cytokine? The cytokine is the chemical messenger to mediate our immune system. What is the inflammatory mediator? What is the meaning of inflammation? Inflammation is or produce the induration or puffing or bulging of our and recruit to the more number of cells. Next, the basic explanation, the immunoglobulin. This is the common structure of all the immunoglobulin is the antibody. It is already known. There are the two chain, one is called the heavy chain and the light chain. There are the two region, one variable region and a constant region. Next is the type of antibody isotype which have the different role in the immune response. These are different isotope of immunoglobulin. Each immunoglobulin has its own function in our body. For example, the IgG is a large amount are present in our blood. Example, more than 80% of the antibodies is IgG type, and the IgM is contained only the 5 to 10%, and the IgA nearly the 5 to 7%, and the IgD less than 5%, and the IgE less than 1%. These are the different isotopes of immunoglobulin. Next, moving on, cytokine response. The next is, this is also released by the host cell in response to the presence of viruses. In a typical scenario, a virus infected cell will release the cytokine. This is called the interferons, causing nearby cells to highlight their antiviral defense system. Cytokines are crucial for fighting of infections and in other immune responses. However, they can become dysregulated and pathological in inflammation and trauma. Next, moving on, course of COVID-19 infection and cytokine storm. What is cytokine storm? The over secretion of cytokine can trigger a dangerous cytokine syndrome. Dangerous cytokine storm syndrome. The cytokine storms are also suspected to the main cause of death in the 1920 and the Spanish flu pandemic. In 1918, the Spanish flu caused around the world the 5 million people are died. In India, nearly the 1.5 crores people are affected in the Spanish flu pandemic in 1918. From this picture, you can very well understand what is cytokine storm and how it is damaging the healthy cell, which leads to the fatal condition. In the case of COVID-19, a severity of illness is divided into 3 stages. The first stage is the asymptomatic, that is viral response phase. The second stage is non-severe symptomatic, and the third stage is the severe respiratory inflammatory stage, that is the hyperinflammatory phase, that is cytokine storm. But now the question is, what is cytokine storm and how it forming in our body? When the virus enter into our normal immune response triggered, and the macrophages and the dendritic cell release the cytokine signal, which leads to inflammatory reaction and start to destroy the invading virus, and it leaves the inflammatory markers, which in turn destroy the lymphocyte while it try to destroy the virus. Then this damaged lymphocyte start to release ever more cytokine, which affect the immune response cycle. By destroying the feedback control mechanism, as a result, more inflammatory reaction will take place and which will destroy the healthy cell as well. So, which leads to acute lung damage. The acute lung damage in turn leads to death. This is mechanism happening in our body when pathogens enter in our body. Next, moving to the complement cascade and its function. Already I mentioned complement is a important soluble factor. The complement contains the more than 25 different type of protein in our blood system. There are the 3 type of complement pathways. One is called the classical pathway, alternative pathway, and lectin pathways. And the classical pathways activate depends on antibodies, and the alternative pathways the independent to the antibodies, and the manus binding lectin pathway activate the manus binding protein. All 3 pathways activate in this year C1 to C9. What is meaning of the cascade? Cascade means one protein activate the other is called this cascade. The complement components that activate into the C1 and the merging into them C3 and the ending into them membrane, forming the membrane attacking complex. The membrane attacking complex, the making the pore and lies the bacterial cell, mainly the extracellular pathogen. The complement system consists of a number of small protein which cleave the specific protein to release the cytokine and initiate the amplifying cascade of further cleavage. The end result of this complement activation is stimulation of phagocytosis. And if you see the functions of complement cascade, first one is called the lysis. Lysis means the membrane attacking complex and making the four and the imbalance of the cell. Example, the release the imbalance of osmotic regulation. There is no osmotic balance. The cell is lysed. Second function is opsonization. What is the opsonization? Or the first one, what is opsone? Opsone is the coating, the protein or soluble factor coating on the pathogen. The process is called the opsonization. The opsonization leads to a phagocytosis. The opsonization, the EC of the phagocytosis. Third one is called the activation of inflammatory response. The already mentioned the inflammation or cause the induration of your immune system. And the third is called the clearance of immune complex. What is immune complex? Immune complex means the aggregate of our antibodies, mainly the IgG and the IgM. After any pathogen enter, the IgA antibodies and the IgM antibody, IgG and IgM antibodies combine with the pathogen to forming the immune complex. The immune complex are clear the complement system. The next moving on, inflammatory response. The inflammatory response is the important response of innate immune system. Usually, the mast cell which present in the connective tissue contain the granules rich in the histamine. These cells will detect the injury to nearby cells and release the histamine, initiating the inflammatory response. Later, when the histamine level increases in the blood, it will follow to the wound site, bringing the phagocytos and their immune cell that neutralize the pathogen. As a result, the blood flux causes the wound to swell, redden, and become warm. The inflammatory response mainly responsible for IgE. That's why the IgM level less than in our blood, less than 1%. Now we have the done with the molecule responsible for the immune response and the pathogen enter in our body. Next, moving on, the cooperation between the innate and adaptive immune response. How the innate immune response and adaptive immune cooperate and improve our immune system. Now we are moving to the most interesting point, that how the innate and adaptive immune response are responding to the pathogen and whether they have working together or not, and whether the innate immune response have impact over the better response the adaptive immune response. If you see this picture, you can very well understand that the innate immune response enhances the adaptive immune response to be the more effective by producing the molecules which trigger the immune response. Next, moving to the adaptive immune response to infections. There are the different stages in the infection. This is the most important step in which should understand that how the adaptive immune response is developed with respect to the infection. When the pathogen enter in our body and reach the cell, it reaches the antigen presenting region, that is the T cell. Then the T cell will be triggered by the dendritic cell, produce the cytokines, and this will kill the pathogen. Of these, T cell will get activates and get replicated, the produce them n number of cells. Then this replicon will get differentiated into the different type of cells, that is effector cell and the memory cell. The effector cell produce the cytokine to kill the invaded pathogens, and the B cell will be stored as the memory cell. This is how the adaptive immune response is developing in our body with the response to infections. Let's move on. The major histocompatibility complex and antigen presenting cell, that is called the MHC. The another factors which trigger the immune response is the MHC, that is the major histocompatibility complex. These complexes are found on all nucleated body cells, and the MHC class 2 are found on macrophages, dendritic cell, and B cell. The antigen binding cleft of MHC1 is formed by the domain alpha 1 and alpha 2. The antigen binding cleft of MH2 is formed by the domains of alpha 1 and beta 1. Next, move on. Antigen processing 1. that is a protein molecule on the left is digested by the cell, and a fragment shown in green is loaded into the MHC molecule that is then displayed on the surface of the cell on the right. Oriented show that it can be scanned by the T cell. The MHC molecule is less than 7 nanometer tall above the cell membrane. Not to scale, which was described by the Javagar Swaminathan and MST staff at European Bioinformatics Institute. The next move on antigen processing 2. In this case, internally produced and externally captured proteins are loaded onto MHC molecule inside the antigen presenting cell and APC. Internally, produced proteins are presented by MHC class 1 molecule, which are found on all the cells in the body. Externally, acquired proteins are presented by MHC class 2 molecules on the specialized antigen presenting cell or APC. Once loaded, the molecules are exported to the cell surface. The next moving on antigen presenting cell. The antigen presenting cell, everyone may be wondering what is antigen presenting cell? It is nothing but macrophage engulf and digest the foreign bacterium, and antigen from the bacterium is presented on the cell surface in conjunction with major histocompatible complex MH2 molecules. In the lymphocytes of the adaptive immune response interact with antigen embedded MH2 molecules to mature into the functional immune cell. Next, moving on, MHC, major histocompatible complex and the T cell receptor interaction. Next, the MHC is interacting with the T cell receptors. That is, if you see the picture, it shows that the crystal structure of interaction between the TCR, the alpha and beta chain, and the human MHC. Example, HLA, human leukocyte antigen. The peptide antigen can just be seen at the interface between the two receptors, the influence of peptide class 2 presentation. The next moving on, the T cell receptor and T cell selection. The adaptive immune cell, like T cell, play the critical role in protecting your bodies against the invading pathogens, a task that relies upon their ability to recognize the pathogen as foreign or non-self. This backs the question, though, how adaptive immune cell distinguish between the self and non-self? How it is that T cell know to attack and kill the invading bacterial cell while leaving our neighboring cell clones unharmed? That answer this question lies in the process of positive and negative selections. When a TCR binds an antigen and MHC complex displayed by the sick or infected cells, the T cell can induce the cell death is called the apoptosis in the top in order for mature antigen recognizing the T cell to develop without being the self-reactive causes and autoimmunity. What is autoimmunity? The autoimmunity is the disorders of our immune system, the autoimmunity cause of the own cell. That the T cell must go through both positive and negative selection. In the positive selection, the T cell in the thymus that bind moderative to MHC complex receive the survival signals in the middle. However, the T cell whose TCR bind too strongly to MHC complex and will likely be a self-reactive or killed in the process of negative selection in the bottom. Next, moving on, the classes of the T cell. Based on this mechanism, the T cell are classified into different types, that is helper T cell regularly. The type is that is called the helper T cell, regulatory T cell, and cytotoxic T cell. This table shows that how these cells are activated and what is its a function. For example, if you see the helper T cell having the CD4 at the surface, what does the CD means? the surface markers, which will activate the antigen presenting cell in association with MHC molecules and activate the macrophage and the natural killer cell in the NK cell. The next, the activation and differentiation of the helper T cell. The pathogen enter into our body and the pathogen are attached to the MHC class molecule forming the MHC complex. The further this T cell helper cell is differentiated into different type. Among the one is called the memory T cell. Next is activation and differentiation of the cytotoxic T cell. If you see them, another type of T cell, that is cytotoxic T cell. These cytotoxic T cell are activated by the antigen presenting MHC molecule on the affected body cells. Once activated, the cytotoxic T cell release the perforin and the granzyme that invade the infected cell and induce controlled cell death and apoptosis. The next, moving on, how the CD4 and the CD8 cell are respond to any pathogen. When the naive CD4 cell, naive means undeveloped or uninfected CD4 is called the naive cell. The naive cell engage the MH2 molecule and antigen presenting cell APC and they become activated. The clones of activated T cell in turn activate the B cell and the CD8 cells, which become cytotoxic T cell. The cytotoxic T cell kill the infected cells. Next, the NK cell means the natural killer cell. What is the function of the natural killer cell? The figure shows, hope you all remember the word I said, natural killer cell. If you see, the role of natural killer cell is immune response. If the cells recognize the MHC1 receptor which is present on the healthy cell, it will not kill the healthy cell, but in case of infected cell, MHC1 will be absent and the cells will lysed. Next, moving on, super antigen and unregulated activation of the T cell. What is super antigen? The super antigen is the bacterial product, and the importance is called the minimum level of the protein or induce the maximum response. That's why it's called the super antigen. The super antigen is mainly present in the Streptococcus pathogens. And the most important point here to noted is the super antigen, which unregulate the T cell, so which turn produce the more cytokine, which leads to death. The next, moving on, affinity maturation. How the mature the B cell, how the mature B cell maturation. Sometime with respect to the affinity towards the antibody plays the vital role in immune response. That is, antibodies that fit quite well are selected early in the immune response. The antibody receptor, the BCR, is mutated within the daughter cell. Many of these mutations bind to the antigen worse than the parent antibody, and the cell the producing these antibodies die. Some antibodies binding the antigen better than the parent, and these cells are live. Next process, the T cell dependent and independent activation of the B cell. The first figure is independent activation of the independent activation. Second one is called the dependent activation. When I said earlier, the T cell will get differentiated into B cell and memory B cell in case of adaptive immune response. These B cells are not always depending on the T cell to get activated. Sometimes it will get activated by itself and segregate the antibodies IgM. The next, the immunological memory. The immunological memory is the important part of the immune system, especially in the adaptive immune response. Here and there, I used the word memory cell, but never discussed how these memory cells are formed in our body. That is, when initially binding an antigen to the B cell receptor BCR, a B cell internalize the antigen and present it on MH2. A helper T cell recognize the MH2 antigen complex and activate the B cell. As a result, memory B cell and plasma cell are made. The plasma cell produce the antibodies. This memory cell will get stored in our body for the further use. Next, moving on, rapid and dramatic antibody response. We may wonder, what is the role of memory cell? Right? The answer is that in the primary response to infection, antibodies are secreted first from the plasma cell. Upon re-exposure to the same pathogen, example, the chickenpox, the memory cell differentiate into antibody secreting plasma cell that output a greater amount of antibody for a long period of time. Next, the antibody responsible for vaccination. I hope everyone aware that why we are given the vaccination and what is importance in immune response. The vaccination are the small fragment or non-virulent form of pathogen that will be introduced in our host. Our immune system will react to that cells and act as a result of memory cell will be stored. That is primary response. And then sometimes we will give the booster, the immune response which produce against this secondary response that will withstand our body for quite long period. Now is the third part is the tuberculosis. With this, I would like to take the tuberculosis as one of the infectious disease to understand more how our immune system is formed against the Mycobacterium tuberculosis. Why I am chosen this disease is that I have research on the immunology of tuberculosis in the Department of Clinical Pathology, National Institute for Research in Tuberculosis, Indian Council of Medical Research from 2002 and 2008. Coming to tuberculosis, the Mycobacterium tuberculosis causative agent of tuberculosis, and nearly the 1.5 million people are dead worldwide. This is the old pathogen, but it's caused the large damage to our society. The Mycobacterium tuberculosis is the airborne pathogen that enter into their lungs, first encounter the lung alveolar macrophages. The lungs alveolar Mycobacterium tuberculosis is the intracellular pathogen that reside in the alveolar macrophages and multiplies, they increase the number. After the numerous pathogens are produced inside the cell, they burst the cell and moving into their bloodstream, and through the bloodstream are the spread are disseminated to the different organ or different parts of our body. That's why the different type of tuberculosis occur. Mostly the pulmonary tuberculosis affected the lungs and other tuberculosis, meningitis and etc. The tuberculosis, the important vaccine is called the BCG. The BCG used for more than 100 years, but the BCG protect only in the children up to the age of 13. The adult, the BCG is not effective compared to this child. Next, the Mycobacterium tuberculosis infection. So, if you see the tuberculosis is the world top infectious disease killer, now 90 to 95% are containment and is from the granuloma and will lead to the latent infection. What is the meaning of latent? The latent means it is later. At presently there is no symptom but exists in our body, but as the cause the disease in the later time. But sometime that is a 5 to 10% are active disease which will transmit to other as well. In addition to this, sometimes the latent infection will get reactivated and cause 10% risk factor. In tuberculosis, in India or globally, 90% people are infected with the tuberculosis, but only the 10% of people got the disease. Remaining people are in the latent condition. Next, moving on, the containment of the Mycobacterium tuberculosis. When the inflammation occur, the granuloma will form. The granuloma is nothing but the collection of immune cell like the macrophages or dendritic cell and the B cell. Next, the host cell and the environment of Mycobacterium tuberculosis. If you see, then the Mycobacterium tuberculosis enter into our body, reaches the organs The granulome will form and the cells will produce the immune response. As a result, mycobacterium will get eradicated and mycobacterium are completely eradicated and sometimes it will get replicated in our body and cause the more damage. Next, moving on, the immunological life cycle of tuberculosis. In this picture will shows detailed how the immunological response is taking place with respect to the mycobacterium. The mycobacterium tuberculosis is an intracellular pathogen, but mainly the macrophages are the first line of defense against this pathogen. There are the two type of immune system are working, are innate immunity and adaptive immunity, but the mycobacterium tuberculosis, they evade or escape from the both the immune response and cause them severe cost to our the body. Next, moving on. This is one of the important paper published the report regarding in the TB vaccine and its impact. Though many vaccines have been designed against the tuberculosis, the mycobacterium is evading and causing the disease. So, we are not able to completely control the tuberculosis. Next, moving on, need for the TB vaccine research. The WHO has the plan to eradicate TB completely by the 2015. So we all should join our hands to do so by doing the following research. That is more fundamental research to the taken over. The new drugs and the new vaccines should be designed in order to eradicate the TB completely by the 2015. But next, moving on, the tuberculosis and COVID-19. The recent studies show that the COVID-19 have more impact on the TB because TB patients are most vulnerable to infectious disease, including the COVID-19. So they should given the special attention to save them. Next, moving on, herd immunity. The herd immunity is a popular term. Nowadays, the herd immunity is a popular term. With all these points, I wish to say that herd immunity is the only solution to all the infectious disease, including the COVID-19. The herd immunity is nothing but when enough that is 60 to 70% people get immunized against the disease, the other person will also protected. If you see, next moving on, the how to boost our immune system. So the only solution is that boost our immune system by taking the healthy food. Next, the immune fitness fit for the future. If you have the immune fitness, our future will also be fitted very well. So to conclude, the immune response to infectious disease are an efficient and effective mechanism against the bombardment of pathogen we face every day. At this point, I wish to state the important quotes by Hans Zinner that the infectious disease is one of the few genuine adventure left in the world. Finally, this slide shows a recap of immunology, that is the serum therapy to the checkpoint control and the pioneer who worked from the amazing immune system. Thank you. Break the chain of infectious disease by avoiding the contacting with that during this COVID-19 pandemic. Stay home and stay safe to break the chain of COVID-19 and have the healthy food to boost our immune system or immune response. Thank you.
Thank you very much, sir, for your effective presentation. The participants can post their questions or clarifications in the chat box. So first question, whether T cells are activating in soil organisms?
The T cell activate the soil organism. I've already mentioned that T cell is the important component of the adaptive immune system. The adaptive immune system well developed into this vertebrate, especially in the mammals only the adaptive immune response are active. In the lower animals, the plants and the other animals, the adaptive immune systems are not evolved.
Second question, sir. Can you suggest diet that help in increasing our immunity?
Immunity is the prime for our body, but the diet, the example in our body is contained the several malnutrition disease. The the immune example tuberculosis, example the HIV. The HIV is not a serious disease. HIV is a weakest pathogen in the world, but is a damage or control or reduce the T cell. That is the problem. The T cell is the important to adaptive immune system. The reduce the T cell number is the indication of the AIDS. Generally, the category is different. Globally, less than 500 CD4 cell is called the AIDS condition. But in India, the less than the 300, the CD4, the CD4 count is less than 300 is called that causing the AIDS disease.
Why it's difficult to design the drug or vaccine for infectious diseases?
This question is the starting from this evolution. Based on this Darwin, the Darwin theory, there are the two parts. One is called the there are the two type of development. One part is pathogen and the other part is our immune system. The pathogen also evaluate and our immune system also evaluating the competition between the pathogen and our immune response. That sometimes the pathogens are evade, the pathogen developed several evading mechanism or escape from the immune system. That's why our the vaccine targeting or drug targeting is critical.
Why the adaptive immune response not working for COVID-19?
Adaptive immune response is mainly there are the two type of immune system. One is called the innate immune system and adaptive immune response. The adaptive immune response, as I already mentioned, in the cytokine storm. The cytokine storm is mainly the cytokine. The cytokine is cytokine is called the chemical messenger. The chemical messenger activate both the macrophages and the T cell. The T cell, an activated T cell, release the number of interleukins, and the interleukins and stimulate interleukin are controlled in COVID-19. I have not the clear.
Okay, sir. Thank you. Now I invite Dr. V. Mayyalagan, Assistant Professor of Zoology, to propose the vote of thanks. Sir, please.
Sir, am I audible, sir? Sir, am I audible? Hello?
Yes, sir. Audible.
Mayyalagan, sir.
Audible.
Sir. Sir, thank you. Most distinguished President Dr. M. Aruchami, sir. Most respected Secretary and Director Dr. C. A. Vasiti, Madam. Respected Principal Dr. M. Lakshmana Swami, sir. Respected Dean R&D Dr. S. Balswami, sir. Respected CEO Dr. V. Chinnuswamy, sir. And dear participants, very good morning to one and all. On behalf of Kongunadu Arts and Science College and the organizing team of this webinar series, it's my immense pleasure to propose vote of thanks. At this juncture, I would like to thank the convener of this webinar series, Dr. D. Vijay Lakshmi, Assistant Professor of Mathematics, and Dr. M. Vigneshwaran, Assistant Professor of Mathematics, for coordinating this webinar series. I extend my sincere thanks to the organizing secretaries, Dr. M. Revati Bala, Associate Professor and Head, PG Department of Commerce, Dr. N. Senthil Kumar, Associate Professor of Computer Technology. At this juncture, I would like to thank also the today's resource person, Dr. S. Manivannan, Assistant Professor of Zoology, Kongunadu Arts and Science College, for his informative talk on infectious disease, immune response to infection. I also express my sincere thanks to all the participants from various colleges and institutions for active participation. Last but not least, I thank all the technical team members and their support to conduct this webinar series successfully. Once again, I thank you all. Thank you very much.
Thank you, sir, for your nice words. The participants are requested to join us by 11:45 a.m. for the second session. Will be handled by Dr. L. Vidyarani, Assistant Professor of Mathematics on topology. Thank you.